this lecture, we are going to discuss about two most important and powerful techniques for determining three-dimensional structure of molecules, maybe large or small, or organic molecules or macromolecules. These techniques are nuclear magnetic resonance spectroscopy and X-ray crystallography methods. One technique where we can do in nuclear magnetic resonance spectroscopy, the three-dimensional structure could be solved in solution state, whereas in X-ray crystallography studies you require crystals for determining the three-dimensional structure. Now, we will without going in too much of detail, we will try to understand these two techniques, the basic concepts of these two techniques. So, let us start with nuclear magnetic resonance here. NMR, or we can say simply NMR. So, NMR relies on the ability of atomic nuclei to behave like a small magnet and align themselves with an external magnetic field. So, when irradiated with a radio frequency signal, the nuclei in molecule can change from being aligned with magnetic field to being opposed to it. Now, this is called nuclear, for instrument works on stimulating the nuclei of the atoms to absorb radio waves, and the energy of the energy frequency at which this occurs can be measured and displayed as an NMR spectrum. So, this phenomenon is based on the fact that nuclei of atoms have magnetic properties, like a spinning charge acts as a tiny bar magnet, and it has a particular angular momentum, and this could be utilized to yield chemical information. Quantum mechanically, subatomic particles like protons, neutrons and electrons have spin, and in some atoms, for example, say 12 C or 16 O or 32 S, these spins are paired and cancel each other out. In certain other so they will not have any overall spin. In certain other ones like 1 H, 13 C, 31 P, 15 N or 19 F, the nucleus does possess an overall spin, and to determine the spin of a given nucleus, one can use certain rules, like say number of neutrons and number of protons, if both are even, nuclei has no spin. If the number of neutron plus the number of proton is odd, then nucleus has a half integer spin, and if the number of neutrons and number of protons are both odd, then the nucleus has an integer spin, that is 1, 2, 3 like that. So, those atoms which has half spin, that is plus half or minus half, can be utilized in NMR technique. So, if you see in figure here, there are two, like say we can take example of proton. When there is no field, they are like not so much separated, they are just oriented randomly, but when external field is applied or external magnetic field is applied, then there are two species, one having spin plus half which align with in parallel to the applied magnetic field, and there is minus half which align negative to the applied magnetic field. This particular one, one is opposed, that is anti-parallel, and other is parallel as we say, and they are referred to differently for this purpose. The rotational axis of the spinning nucleus cannot be oriented exactly parallel or anti-parallel with the direction of the applied field. It is mostly like what we say, it presses about the particular field at an angle, which is angular velocity or angular momentum, and this particular property or magnetic moment of is measured in terms of angular momentum. The orientations that a nucleus magnetic moment can take against or external magnetic fields are not of equal energy, and spin states which are oriented parallel to the external field are lower in energy than in the absence of external field, and in contrast spin states whose orientations oppose the external field are higher in energy than in the absence of an external field. So, what you have is the two states which have particular spins with plus half or minus half, they are oriented in a particular manner when applied magnetic field is there. Energy separations exist in certain way and these induce the transition between the various spin states, like by irradiating the nucleus with electromagnetic radiation of the correct energy, which is in radio frequency range. Nucleus with no low energy orientation can be induced to transition to an orientation with higher energy. Now, absorption of energy during this transition will form the basis for NMR method. So, as we understand that when applied magnetic field is there, these nuclei have plus half and minus half and they align or are opposed to the applied magnetic field, and when an external electromagnetic radiation most in form of radio frequency or radio waves is applied onto the sample, then the absorption will occur, which will make a induces a transition to an orientation with a higher energy of a particular nuclei from the low energy, and this is the basis of NMR method. This resonance is taken and this is recorded. Now, it is very important to understand, like say for example, in proton NMR, how the particular proton in a certain chemical environment is detected, and that those particular differences which are obtained, these are called chemical shifts actually, and they are taken in with an standard which is TMS standard. Let us discuss that. The chemically different protons which are present in different electronic environments. So, what happens is their differences in the electronic environment will cause the protons to experience slightly different applied magnetic fields owing to the shielding and de-shielding effect of the induced electronic magnetic fields. Over the years, the NMR spectra have been obtained on every conceivable organic molecule in nature or synthesized in the lab. So, in order to standardize the NMR scale, it is necessary to set a 0 reference point and to which all protons can then be compared. The standard reference that has been chosen is tetramethylsilane or TMS, and this compound has 4 CH3 methyl groups single bonded to a silicon atom. So, all of the protons on the methyl groups are in the same electronic environment, therefore, only one NMR signal is generated. So, the electronegativity of all carbon atoms is actually higher than the silicon atom to which they are bonded. So, here tetramethylsilane is taken as the reference molecule, that is it is considered as 0. Now, result in the sigma electrons being shifted towards the carbon atom in the methyl groups, and consequently the protons will be heavily shielded, causing the one signal to be generated at a very high magnetic field strength setting. It is that signal that all other NMR signals of a sample are referenced to. So, this association with the reference signal is called the chemical shift, and the shift is measured in terms of parts per million. So, in very simple terms, tetramethylsilane or TMS is taken as reference, and all other measurements or the protons in different environments will be calculated. The positions will be calculated in the spectra as per the reference point, and these shifts which occur in reference to tetramethylsilane are called chemical shifts, which are measured in parts per million. Now, after the sample have been referenced to the TMS resonance at 0 ppm for actual NMR peak position in hertz is divided by the resonance frequency of the spectrometer, which is in megahertz. Thus, 1 is dividing hertz by megahertz, which is a part per million. So, 1 ppm on a 58 megahertz NMR instrument is actually 58 hertz from the resonance position of TMS, while on a 300 megahertz NMR instruments 1 ppm is 300 hertz for the TMS resonance position. So, these are the differences here. Now, with the standardization and normalization in place, one can always unequivocally say that all benzene protons will resonate at around 7.16 ppm, no matter what instrument is being used in the analysis. So, there is like in this here you have a down field and there is a up field, like you have a TMS which has a reference and you have different positions which proton will possess in different kinds of molecules, as you can see alkane, alpha H, water, aromatic, aldehyde. Depending on the chemical environment, these peaks will appear and they will be from it is taken from 0 to 10 ppm in here. Now, NMR instrument we like here TMS standards available to the reference online spectrum. So, the NMR peaks in the spectrum itself are used to reference the whole spectrum based on a well established knowledge of the process stream chemistry. The various aromatic groups in the spectrum can be picked and assigned to their known chemical shift values that have been logged in large database of NMR chemical shifts. So, there are databases available and chemical shifts could be directly seen from or could be compared to those database even if you are directly doing it. So, proton NMR chemical shifts for a common functional groups if we see, and if we compare here you can see that depending on like different chemical environments, the protons will show different shifts actually or different chemical shifts, like starting from the 0 ppm TMS. If it is a saturate H, it is somewhere around very close to the TMS, and as the environment chemical environment changes, you can see the shift in the chemical, and which is highest around for carboxyl group in here, proton in carboxyl group, then it is aldehyde group, and finally aromatic CONH2 group, and likewise it is like least here for RNH2 or others. So, or saturated H will have the least chemical shift. So, what is the basis here of all those things here? So, electronegative groups are here de-shielded and tend to move NMR signal from neighboring protons further downfield to higher ppm or you can say to higher ppm values. Now, protons on oxygen or nitrogen have highly variable chemical shifts which are sensitive to concentration, solvent, temperature. So, all these things will play a role in chemical shift. The system of alkenes, aromatic compounds and carbonyls strongly de-shield attached protons and move then to higher ppm values. There are 6, if we compare, if we see the NMR experiment, there are mainly 6 stages involved in structure determination by NMR. One is to prepare the sample for NMR. So, sample could be done like by proton NMR, it has to be dissolved in proper solvents, and if it does not contain radioactive nuclei like we have discussed like 1H or say for proteins 13C or 15N, then those have to be put in there. Then optimized conditions has to be created for the experiment. After this, the NMR signal is recorded on the machine and NMR spectra is obtained. Now, this NMR spectra once it is obtained on the NMR spectrometer, then these spectra has to be assigned like peaks has to be assigned, that is we call it assigning the spectra. In here what happens is that from like we discussed, different peaks are selected, and these different peaks which which standard values are given in database, they can be picked from there, and according to the standard values which could be compared, the peak assignment could be done. So, that is another important thing. Then compile the list of different for example, if you are doing 1D NMR, it is a simple proton NMR, and it gives identifies different protons in various chemical or electronic environment, but one can perform one dimensional apart from one dimensional, it could be two dimensional, three dimensional spectroscopy. So, various distance restraints and interactions has to be like the 2D or 3D NMR spectra are much more complex, and assigning the peaks is a very complex process in there. Once this whole list of say certain peaks are assigned, then a 3D structure needs to be computed, which should be consistent with the data, and this is 3D structure is done on the basis of different interactions, which is like there are a lot of effects like correlation effects or nuclear Overhauser effects are there, which needs to be taken into consideration. So, when you have to read the spectrum, very important part of the overall experiment is reading the NMR spectrum. So, an NMR spectrum appears as a series of vertical peaks or signals, which are you can say lines or very it is a peaks as we have seen in other spectroscopy technique. They are very fine peaks distributed along the x axis of the spectrum as per their chemical shifts actually, and each of these signals correspond to an atom within the molecules being observed. Now, the position of the each signal in the spectrum gives information about the local structural environment of the atom producing the signal. Say for example, if it is a 13C NMR spectrum, then ethanol will be shown where you have CH3 and CH2, the two carbons in ethanol are in different structural environments, and hence each produces a signal in the NMR spectrum. Likewise, the carbon attached to oxygen is de-shielded due to the electromagnetic nature of the oxygen, and this shifts its signal towards the left in the spectrum. So, this is very important that whether it is a shielded or de-shielded electrons, whereas the carbon bonded only to hydrogens and carbons appears at the right of the spectrum. So, if there is a more de-shielding of there is a electronegative atom, then there is a shift actually, like you can see there are two peaks for two carbons in here, and one which is shifted to the left, which is connected to electronegative atom rather than the other carbons which are simple. So, these are the peaks which are obtained in here, and it depends on their electronic environment for carbon, or say for NMR, if we say proton NMR, where proton acts as the nucleus which is resonating. Then in 1H NMR spectrum, say for ethanol, the two protons of the CH2 group neighboring the oxygen are further to the left in the spectrum, while the hydrogen of the CH3 group that is most remote from the oxygen produces a signal towards the right. So, one which is closer to the oxygen will be having a higher chemical shift as compared to the one which is connected to simply the carbons actually. This is like signals in 1H NMR spectrum do not necessarily appear as a single line, because there is a splitting pattern as we have seen in here, and this gives information as to how many hydrogens are present on the neighboring carbon. So, depending on the split, it will tell about number of carbons, and also integration of the 1H NMR signal allows number of hydrogens in each environment to be determined. So, if you see here in this figure, depending on how many hydrogens, there is a splitting of the peaks occurs actually, and that is will be determined as per the number of atoms attached to a particular groups. So, reading NMR spectrum is a complex process, but just in a very simple way, as we have seen the chemical shifts and as we have seen how a particular atom could be, or a proton molecule as we are doing proton NMR or carbon NMR, that particular radioactive nuclei could be shielded or de-shielded. Depending on that, whether it is attached to a more electronegative or electron withdrawing group or to a simple saturated group, there will be a chemical shift, like it was higher chemical shift for say atom connected to oxygen, and lower chemical shift for atom which is connected to say carbon group, where no such effect occurs. So, accordingly these spectra will be obtained, and according with reference to the database, it could be understood that whether this is present in say environment which is having like say it is connected to carboxyl group, or say simple carbon like in CH3, or it is connected to aldehydic groups, or say other groups. So, standard values your data needs to be compared to the standard values, and peak assignment has to be done. The instrumentation part of this NMR technique, if you see here, this is how this whole instrument looks like. It is a big cylindrical vessel in here, where all the things are available, and if we see in detail here, if you see on your screen, it is like your sample, a spinning sample tube is in here, and it is has a coil which is around this sample. These are sweep coils, and there is a magnet pole, or you can say very powerful magnets in here. There is a radio frequency transmitter which provides the radio frequency, and these are radio frequency receiver and amplifier which will collect the signal. At correct resonant frequency or radio frequency, there will be a signal which is like transition of nuclei from one lower state to higher state, and the applied magnetic field is provided by this, and resonance will occur only when the frequency or the energy matches with the two resonating states, and that particular spectra will be collected and then could be analyzed as we have discussed. This is another representation of the same thing, where is RF pulse generator, you have a rotating anode, and there is a spinning sample tube here. There are two powerful magnets, and there is RF detector which is there, and finally signal is obtained in terms of peaks, and which could be analyzed on the automated system, with lot of different softwares are available for computing the 3D structure from say 2-dimensional or 3-dimensional NMR. 1-dimensional NMR is only for identification of, say proton NMR is only for identification of the chemical environment or electronic environment of a particular nuclei. When a compound containing hydrogen is placed in a magnetic field, say a proton NMR, we say the spin of the nuclei can be aligned parallel to the lines of the magnetic field or to anti-parallel, which is called lower energy is called alpha, and it is represented by alpha, and higher by beta, and the applied radio frequency energy from the generator will cause the spins to be promoted to the higher energy state, and this will be the base you can record the NMR spectra. So, we have not talked about quite lot of things in here, as these are like complex, these are like NMR process or NMR spectroscopy is very complex and detailed discussion is required for this, but as we understand in a very simple way, that is, one is there is a nuclear magnetic resonance, or certain nuclei with half spins, that is plus half and minus half, and like we said nuclei we have mentioned. These when they are spinning around a particular axis and they are precessing, you can say, at a particular angular velocity, and or you can say angular momentum, they act as magnets or tiny bar magnets, and when there is a applied, so they are oriented randomly, but when there is a applied magnetic field, then these will orient or they will be divided into two states. One is plus half, which is aligned parallel to the external magnetic field and which is at lower energy, and there another population will be of with negative spin or anti-parallel to the or opposed to the external magnetic field, and which is again at a lower energy. So, population in the higher energy is less than the lower energy, and as radio frequency or electromagnetic radiation in form of radio frequency waves is like when sample is irradiated with that, then at particular frequency, that is equivalent to the resonating frequency of the nuclei from lower state to higher state, then some of these will shift upwards or into higher state, and this NMR spectra is recorded, which gives information about the electronic environment of that particular nuclei. So, for example, a proton NMR when we are doing, these environments are calculated in terms of chemical shifts from a reference molecule, that is TMS, and TMS is taken as 0 parts per million, and then as per the shift, chemical shift, which could be farther from the TMS, it is calculated. So, what you have is you like those protons which are attached to say electronegative atoms or other which are electron withdrawing groups, they will have like a shift higher chemical shift as compared to those which are attached to saturated atoms like carbon or others. So, accordingly from database these things could be compared and they could be peaks could be assigned and proper identification can be done. For 2-dimensional and 3-dimensional NMR, the NMR spectra will be obtained in 2 or 3 dimensions, and those NMR spectra will be very complex, and then peak assignment needs to be done. Lot of things are automated, but still manual intervention and expertise is needed to work on these spectra and to deduce the 3-dimensional structure. Now, most of the organic compounds or any compound in solution could be used for could be taken for determining NMR structure. Protein molecules could be solved here, but there is a limit, like very high molecular weight protein like above 30 kDa cannot be done in here. It becomes much more complex, and then proton NMR cannot be done on these complex protein molecules, as signal number of proton signals will be very high to really deal with. So, there is rather carbon 13 NMR or N15 NMR needs to be performed on those to solve the protein structures. So, this was in a very brief about NMR spectroscopy. Now, there are a lot of applications of NMR as we were discussing. NMR has become a sophisticated and powerful analytical technique that has found a variety of applications in many disciplines of scientific research, medicine and various industries. Modern NMR spectroscopy has applications in biomolecular systems and plays important role particularly in structural biology. There are methodologies which have developed it has become a versatile spectroscopic technique for analysis of lot of different kinds of biomolecules and which could go up to very high, like you can have complexes, interactions could be seen in here, and a lot of different analysis could be performed. Together with X-ray crystallography, NMR spectroscopy is one of the 2 leading technologies for the structured for the 3-dimensional structure determination of macromolecules at atomic resolution. In addition, NMR provides unique and important molecular motion and interaction profiles containing pivotal information on protein function, and this information is very critical to drug development and various other applications. Some of the applications which are very important in very short, one is the solution structure which could be done for various kinds of molecule. Then molecular dynamics could be performed for say motional properties of biomolecules. Protein folding could be done, which is very powerful for determining intermediates or in folding. Then ionization states could be determined, that is chemical properties of functional groups in biomolecules, such as ionization states of ionizable groups in active site of enzymes could be determined, and lot of other analysis could be done. Weak intermolecular interactions could be seen in here, and protein hydration to detect interior water and its interactions could be done this one. A lot of this technique could also be used for direct detection of hydrogen bonding interactions, and it is useful in identifying drug leads and determining the conformation of the compounds bound to the enzyme, receptors and other proteins. So, these are like in metabolic analysis also this technique is utilized. So, there are a whole lot of different applications of this, where chemical and different material analysis could be performed in chemistry, physics and biology. So, NMR spectroscopy is a very powerful and very important technique which is utilized in various areas of biotechnology. Let us move to the next technique, that is X-ray crystallography. Now, X-ray crystallography is one method where you require you can solve three-dimensional structure of any molecule provided there are crystals for that particular molecule. So, what you require is you require a crystal of a particular substance or material, and the basic very basic concept is that what you have is that when there is a crystal of a molecule is available, then X-rays hit the crystals, and this crystal causes the beam of X-rays to spread into many specific directions according to the diffractions. Like the X-ray, the crystal is a periodic arrangement of molecules or atoms in the 3 dimension. It is a regular arrangement in the 3 dimension. So, there is a particular kind of diffraction pattern characteristic for each molecule, and from the angles and intensities of these diffracted beam, a crystallographer can produce a 3-dimensional picture of the density of electrons within the crystal. And from this electron density, the mean positions of the atoms in the crystal can be determined, and with certain knowledge their chemical bonds, disorders and other information as per the data is obtained. Crystals, if you say, they are made of infinite number of unit cells, and unit cell, if we say, it is the smallest unit of a crystal, that is the repeating unit of the crystal, and it repeats in all 3 directions and will generate a whole crystal. So, if I say what is a 3 unit, if I say this is like, say, just for example, then it has a crystal or a unit cell in a crystal, which like it will be made like in all direction, there will be lot of unit cells which will make a crystal, and it has 6 main things. One is 3 axis, x, y and z, or a, b, c you can say, and there are so we call it in this in proper crystal it is a, b, c, and there are 3 angles alpha, beta and gamma between the 3 axis actually. So, if we say we are not going too much detail, but crystal, there are crystal systems which are grouped into 7 crystal systems according to the characteristic symmetry of the unit cell. So, symmetry is very important part of crystallography, because that is the basis for determining the crystal the structure of the molecule, as particular symmetry will provide particular type of diffraction, and that gives a lot of information and all different mathematical like Fourier transform thing is based on in a part based on symmetry actually. So, the characteristic symmetry of a crystal is a combination of many one or more rotations and inversions, and what you get is different types of what I was calling crystal systems, which are these. These are, if you see, these are cubic, monoclinic, orthorhombic, tetragonal, triclinic, then trigonal and hexagonal. These are crystal systems which are obtained, just to give you an idea. And then from this combination of all available symmetry operations, there are 32 point groups and with translational symmetry and within the all available lattices, which are 14 Bravais lattices, leads to 230 space groups, and these are described and arranged in you can say that space groups that describe the only ways in which identical objects can be arranged in an infinite lattice. So, that is a very important part. These are the only possible space groups actually to be arranged in here. There are a lot of symmetry operations which could be rotational, inversion, reflection, and there are a lot of lattice nominations which could be started like reference point from 0 0 and axis directions, which are we have shown A, B, C, and these lattice points could be shown in here. Like if you see, there is a 0 0 reference point, and on all directions you can say like they will be given certain. So, in a unit cell could be divided into different lattice points at different directions, and positions will be defined by those points actually, which are in, like if you can see, this is divided into 0 0 1, 0 0 2, here it is 1 0 2, and here it is like in another direction it is 0 1 0. Depending on that, these things are like we are not going into detail, but just to give you an idea. Lattice planes are defined using Miller indices and calculated as reciprocal of the intercepts of the plane on the coordinate axis. So, the plane which might contain 1 0 0, 0 1 0, 0 0 3. These are three axes actually. Miller index is a series of co-prime integers that are inversely proportional to the intercepts of the crystal face or crystallographic planes within the edges of the unit cell. So, it describes the orientation of plane in 3D lattice with respect to the axis. So, general form of the Miller index is HKL, where H, K and L are integers related to the unit cell along the ABC crystal axis, as I have shown you. So, this was little bit about symmetry, and for detailed study one should go deeper into it. Now, X-rays were discovered like in 1895 by Röntgen, and likewise there was like crystal symmetry studies were being concluded at that time, and the field of X-ray crystallography of biological molecules took off like after long period, like with Dorothy Hodgkin with solving the structures of like vitamin B12, cholesterol, penicillin, etc., and she got Nobel in 64 for this kind of work. Crystal structure of proteins which are hundreds of times larger than smaller molecules started with late 1950s with myoglobin by Max Perutz and John Kendrew, and which they were awarded Nobel Prize in Chemistry in 62. So, basic principle on which this X-ray analysis or X-ray crystallography works is on the Bragg's law. So, if we consider the crystal here, if we see the molecules which are arranged in different planes actually, if you see these are molecules, or you can say atoms, these are diffraction planes which are arranged in here, and this is the distance between the two planes or diffracting planes. So, when X-rays hit the plane, then it is diffracted, or you can say reflected here, and this is at a particular angle which is like this, and then when it is diffracted on another plane, then this particular one could be related by this whole there will be you can say phase difference will occur, and so there will be constructive and destructive interferences occur, and this whole relationship is given by 2D sin theta and lambda, that is the Bragg equation. So, here like as we explained, d is the spacing between the diffracting planes, theta is the incident angle, n is simple integer, and lambda is the wavelength of the beam, which is for X-rays like copper one, which is generated from copper anode, is 1.54 angstrom, which is very similar to the bond angles. these specific directions appear as spots on the diffracting patterns, what we got reflections actually. So, X-ray diffraction results from an electromagnetic wave, that is X-rays, impinging on a regular array of scatterers, that is repeating arrangement of atoms within the crystal. And Bragg's law here, interference effects are observable only when. So, according to the Bragg's law, interference effects, which is could be both constructive or destructive, these are observable only when radiation interacts with physical dimensions that are approximately the same size as the wavelength of the radiation. So, only diffracted beams that satisfy the Bragg conditions are observable, which is constructive or destructive, and diffraction can thus be treated as a selective reflection, and n is an integer which is an order, lambda is the wavelength of the radiation, and d is the spacing as between the lattice planes, and theta is the angle between the incident and reflected beam, and incident or beam and the lattice planes actually. So, X-rays for chemical analysis are commonly obtained by simple rotating anode generators, which could be in-house generators, or it could be synchrotron radiation facility. In rotating anode generators, a rotating metal target is bombarded with high energy electrons, and that knock out core electrons. An electron in an outer shell fills the hole, as we have discussed earlier in X-ray spectroscopy introduction, and then inner shell and emits the energy difference between the two as an X-ray photon. Now, as X-rays are diffracted by electrons, so just to understand that molecules or atoms are arranged in crystals, and there is an electron environment around each atom. Atom is made up of proton, neutron and surrounding electrons. So, X-ray diffraction occurs due to the electrons, and this is a elastic diffraction, and due to the constructive and destructive interferences, one will get the intensity patterns due to, like, if there is amplitude are in phase, then you will get high intensity. If there is not in phase, then low intensity spots, and this this set is produced, and this X-ray diffraction data set produces an electron density map by Fourier transform method. Why Fourier transform? One of the very important part of X-ray crystallography is phase solution, as we will discuss. Instrumentation for X-ray diffraction, if we consider, is very simple. One is that X-ray source, which could be like synchrotrons radiation source or rotating anode tubes, as we have discussed. Now, X-rays are used to produce the diffraction patterns because their wavelength is typically the same order of magnitude, 1 to 100 angstroms, and spacing between the planes in the crystals. The X-ray scattering is determined by the density of electrons within the crystals. So, more higher the packing order in the crystal, or they are more densely packed, higher will be the diffraction. And since the energy of an electron is very much greater than that of valence electron, the scattering may be modeled as Thomson scattering, the interaction of an electromagnetic ray with a free electron. So, major aspect here, if we go through the procedure here, what you have got is the crystal, and crystal X-rays are focused on the crystal, and the X-rays are diffracted by the crystal. What you get a diffraction pattern in forms of intensity. This diffraction pattern is a characteristic of that particular molecule, and this so what we are doing is, like in microscope, what you have is objective and eyepiece, and finally you see the image, but in here there are no lenses to focus the X-rays. So, what is done is the pattern is recorded in between actually, and this pattern, which is like not focused and image formation is not taking place. The image formation is done by going backwards through Fourier transform method, and Fourier transform finally makes an electron density map, which is we call phase solution actually, and the problem is called phase problem. Electron density map is the map of the electrons surrounding the atoms. So, the electron density map, and with the knowledge of the molecule you can generate this web like structure which is electron density map, and finally, from electron density map atomic model or the atomic positions, and from that whole structure is derived. So, procedure as we have discussed is very simple, where you have a single crystal. X-ray crystallography has 3 steps. One is most difficult and the bottleneck is to get good diffracting crystals. So, adequate crystals has to be prepared from the material which needs to be analyzed. Crystals should be sufficiently large, say 0.1 millimeter or above, and they should have homogeneous composition. They should be regular in structure, that is good diffracting crystals with no significant imperfections. Second step is the crystal is placed in intense beam of X-rays like single wavelength, which is monochromatic X-rays producing the regular pattern of reflections, and the crystal is rotated at 1 degree or so, so that reflections appear like many frames are collected at different like angles, so that every like all symmetry it is collected, all full data is collected as per the symmetry operations. For determining the symmetry, first single image could be collected, and from diffraction data symmetry of or a space group could be calculated, and then one can decide how many reflections or how many frames, like whether it has to be rotated to 180 degree or like as per the space group the number of reflections will be or number of rotations will be performed. The intensity of every spot is recorded at every orientation of the crystal, and multiple data sets may have to be collected, with each set covering slightly more than half of full rotation of the crystal and typically containing tens of thousands of reflections. So, remember as per symmetry operations, actually you can determine the number of reflections, and once you know the space group, then say depending on if it is a cubic symmetry, then if you do 180 degrees you are repeating the data, but if it is a triclinic, then you need to collect in full at 180 degrees with 1 degree rotations, the crystal data has to be collected. In the third step, the data are combined computationally with complementary chemical information, and to produce and refine a model of the arrangement of atoms within the crystal. The finally refined model of the atomic arrangement is called crystal structure, and it is refined to a particular value which is acceptable, which is called R factor or reliability factor, and it is comparison of observed data and the experimental data. Now, crystallization, which is very important, or bottleneck, you can see crystals looks a very simple, it is a depiction of how crystals might look like. There could be different shapes, sizes of the crystals, they could be various types of, they could be thin sheet like, or they could be rectangular, or different shapes could be obtained. To obtain the crystals, there are various methods, which is hanging drop or sitting drop or dialysis methods. Now, if you can see here in the figure, in hanging drop, this drop of the say protein or any material is hanging, and this whole chamber is sealed, and because of these precipitants which are at higher concentration than in the drop, there will be evaporation of from the precipitant from the protein drop to precipitant. So, crystals will occur. It is like you are taking the protein solution or any analyte solution to supersaturation, and in that process two things can happen. One is protein or things could precipitate, or they could form crystals. So, that is a very tricky situation, and many times it is very hard to get crystals. This is hanging drop, which is here named because drop is hanging. Then there is a sitting drop, where a drop is put in here, and this is precipitant or reservoir solution, and exchange of solvent occurs in here. Again this is brought to supersaturation by keeping it for some time, and crystals might form in here. Then there is a dialysis method, where through a filter, a membrane, which is dialysis membrane, the solution or the protein and solution or reservoir solution or precipitant is like they are interacting, and due to difference in their conditions, that is precipitants conditions, water will be extracted out from the protein solution and will be brought to the higher, or you can say supersaturation stage to yield crystals. So, these are all methods, but there is no definitely one cannot say that crystals will grow or not. This is like you have to, it is a hit and trial method, and many attempts with different conditions has to meet, like with concentration differences or pH differences, it has to be performed. So, crystal growth is characterized by, like say, nucleation of a microscopic crystalline possibility having only 100 molecules, followed by growth of the crystal, and then diffraction quality crystals are obtained, and that is the main aim of the crystallographer, to obtain a good quality crystal. So, many times you have to screen, or there are a lot of screens available in the market to like put your solution, put your protein or analyte, and in different conditions, like it could be 150 or maybe more types of conditions are there in a particular screen, and possibly you might get a crystal in one of the conditions. Once crystal is obtained, the crystal is in the drop, and this crystal is picked up, either it could be mounted in a loop or in a capillary. So, this is picked up by surface tension, it could be picked up, the crystal comes in the loop, then it could be frozen with liquid nitrogen, or you can do it with liquid propane and then can put in liquid nitrogen. This could be stored also for a long time in proper condition. So, once the crystal is picked, this could be put in front of the X-rays, and then data could be collected. Now, capillary or loop, which is like once crystal is in capillary or loop, this will be mounted on a goniometer head, and this goniometer allows it to be positioned accurately within the X-ray beam and rotated. So, you can position in front of the beam and properly center your crystal, so that it is bathed by it is in X-ray direction. Since both the crystal and the beam are often very small, the crystal must be centered within the beam, so that within 25 micrometer accuracy, and this would aided by, there are camera and there are systems available, so that you can do that. Then mounted crystal is then irradiated with the beam of monochromatic X-rays. The brightest and most useful X-ray source are synchrotrons, where even modulation of frequency or modulation of different wavelengths could be performed, and you get better resolution there. They also make it convenient to tune the wavelength of the radiation in synchrotron facility, which is useful for multi wavelength anomalous dispersion method phasing. Synchrotrons are generally national facilities, and there are dedicated beam lines actually for collection of the data. So, what you have is necessarily, if you summarize, there is a X-ray source, the crystal mounted in a loop, it is irradiated, diffraction pattern, the crystal diffracts as per its packing and other parameters you can have. So, it is diffracted. Now, remember higher, like as we have discussed in microscopy also earlier, higher the angle of diffraction, that is more outer diffraction from the center axis. Now, center it is not diffracted. So, as you go along this angle, higher the angle or higher the value, then more will be the resolution actually. This is a screen, or you can say this could be a photographic X-ray film, or it could be a phosphorescence screen as in imaging plate scanner, or there could be other CCD detectors and other detectors could be there. And finally, what you get is the resolution pattern. Now, if you can see, this is the center, and as you go here to the periphery, more is the diffraction towards this side, more is the resolution, that is we get. higher resolution. It means, if the lower is the value, we say of, if we say 1 angstrom resolution, then it is higher resolution. If we get 2 angstrom or 3 angstrom, then it is a lower resolution. So, depending on how much information, that is 2 atoms which are closely spaced, if they could be resolved, then it is the higher resolution, and it is will be obtained when there is a higher reflection or higher diffraction at higher angle actually. Data analysis is done as per the crystal symmetry, unit cell and image scaling. Recorded series of diffraction patterns in 2 dimension, and like I said, many frames are collected and all this data is combined, and this is like through Fourier transform converted into 3 dimensional model of the electron density map, and this is done through mathematical technique called Fourier transform. Each spot corresponds to a different types of variation in the electron density, and these variations corresponds to which spot, or you can say indexing is done, the relative strengths of the spots in different images, and how the variations should be combined to yield the total electron density. All these things are very complex procedural. Now, it is quite automated, and many different advanced methods of phasing and indexings are available, so that better results could be obtained. Now, data processing starts with indexing the reflections, that is, when you have collected many frames at, say, you rotate the crystals by 1 degree, then those it is like you have identified. So, what is done is, here indexing means identifying the dimensions of the unit cell and which image peak corresponds to which position in reciprocal state. So, there is a direct space and reciprocal space. Direct space is the crystal space and reciprocal space is diffraction space. So, a byproduct of indexing is to determine the symmetry of the crystal, that is its space group. Like once it is done, reflection symmetries is formed. There are 65 space groups of 230 possibles which are allowed for protein molecules, and almost always chiral indexing is generally accomplished using an auto indexing routine. Once it is done, having assigned the symmetry, data is then integrated. This converts the hundreds of images containing thousands of reflections or diffraction pattern as we say into single file, and it records the Miller index of each reflection, that is positions in the unit cell. And remember, we are not talking about direct space, it is a reciprocal space. Each reflection and the intensity for each reflection, reflections are in reciprocal state, and finally, full data set will be with hundreds of reflections will be done. There will be lot of repetitions also, which is calculated for degeneracy, and once it is done, then phasing is done. In phasing, data collected from diffraction experiments in reciprocal space is a reciprocal space representation of the crystal lattice. The position of each diffraction spot is governed by the size and shape of the unit cell and the inherent symmetry within the crystal. The intensity of each diffraction spot is recorded, and this intensity is proportional to the square of the structure factor amplitude. The structure factor is a complex number containing information relating to both amplitude and phase of the wave, and in order to obtain an interpretable electron density map, both amplitude and phase must be known. And so, for this, phase cannot be directly recorded during a diffraction experiment, and this is known as the phase problem, as I was talking, that you are collecting data in between. So, the estimates has to be done by various ways, like for small molecules there are direct method available, which is like due to certain information, you can say phase information is like indirect methods is exploited, and for like if the resolution of the data is better than 1.4 angstrom and it is a small method, a small molecule, direct methods could be done. But for bigger molecules, there you have to have either methods are like molecular replacement. In molecular replacement, if a related structure is known, then that could the phases of that could be taken for generating the electron density of your molecule. There could be anomalous X-ray scattering. This is another different method where anomalous X-ray scattering could be utilized. Here X-ray wavelength may be scanned past an absorption edge of an atom, which changes the scattering in a known way, and by recording the full sets of reflection at 3 different wavelengths, one can solve for the structure of the anomalously diffracted atoms and hence the structure of the whole molecule. Then there could be heavy atom methods, that is native crystal data is solved is taken, and then the native structure is soaked with heavy atoms like say platinum or gold or mercury, and then data is collected. The phases of heavy atom are calculated, and then those phases are utilized for solving the structure. So, these are different methods. So, what you get in the end is, if to summarize, this is a crystal, you get diffraction pattern by diffraction by exposure of crystal to X-rays, there is electron density, and finally, you get the model by different phasing statistics. So, finally, like not all the things could be covered both for NMR and X-ray crystallography in here, but in essence X-ray crystallography, the 3 important factors is, one is to get the crystals, then second is to collect the good data, high resolution data, and third is to solve the structure by solving the phases actually, that is to solve the phase problem, as we have discussed with different methods. Once the phases are known of the waves, then the positions of the atoms or electron density could be generated, and electron density maps once are obtained, then specific positions of the atom in the electron density, and with the knowledge about the structure or the composition of the molecule, like say for example, we know the protein sequence, then it will be easier to put in the amino acids as per the sequence, and they could be arranged in electron density and could be compared, like for example, a side chain of aromatic or for say arginine will have longer density as compared to alanine, and those things could be compared in, and then refinement could be performed to get the solution to as close or as reliable as possible, as per there are different methods, different various validation methods for say calculating bond lengths, bond angles, and various other parameters where whether an atom or whether an amino acid is in allowed or disallowed space as per the Ramachandran plot, like phi psi values are taken, and that is how the whole structure could be solved. Lot of things we could not cover because of certain constraints, but this will certainly initiate and make you aware about these 2 powerful techniques to solve the 3-dimensional structures. Thank you.